Thank you very much for inviting me to give a talk today. It is a pleasure to talk at the Annual General Meeting 2021 of the British Lichen Society. Southeast Asia is a known hotspot of biodiversity. The region is uniquely biologically complex, reflecting its complex biogeography and result in zonation of regional biotic patterns and complex biogeographic divides, which require further research to fully understand. IUCN data at a country level shows South Asian countries have the highest global diversity for several taxa. However, the level of taxonomic uncertainty associated with many taxa may be significantly higher than other parts of the world, with only a small portion of some large taxa described. And this lack of knowledge makes regional prioritization challenging. The region is also said to be the most biologically threatened. These threats are complex but need to be understood to develop effective conservation plans. Quantitative extrapolations and molecular data for species complexes in tandem with new fieldwork carried in underexplored areas of the tropics suggest that family Graphidaceae may contain up to 3,500 species, that is 1,400 undescribed species in addition to the 2,100 currently known. The family was traditionally divided into 8 genera based on ascoma and spore types. Molecular phylogenies have intensely changed our understanding of generic classification of this largest tropical crustose family. Recent revisions have substantially changed genus concepts in Graphidaceae, now recognizing 82 genera, including newly described Allographa, Jacota, etc. Further, some more new genera are waiting to be published, mainly collected from South America. Each year, over 50 new species are collected in family Graphidaceae, mainly from tropical regions. I wish to show you some of the main tropical habitats for Graphidaceae. Senior lichenologist Pat Woolsey, who has worked in tropics for several decades, said while she was working in Sri Lanka, these rainforest trees are going crazy with Graphidaceae. She was very disappointed due to lack of Palmiliaceae. Rainforests in tropics are the most complex and species-rich ecosystems in the South Asia. Rainforest trees are mostly covered with crustose lichen species and have hundreds of Graphidaceae recording on them. At least 5 out of 10 rainforest lichen specimens we collected in Sri Lanka are Graphidaceae. Montane ecosystems are found on the slopes of mountains. Temperatures fall as elevation increases, causing this ecosystem to stratify. These forests have very rich crustose lichen diversity. Although trees are mainly covered with foliose and fruticose species, Graphidaceae is one of the most prominent lichen family in Asian montane forest. On naked barks of giant trees in semi-evergreen forest, you can see huge patches of Graphidaceae, mainly Telotrema, Myrotrema, Ocellularia, Leucodecton-like genera. Some of these patches are more than 1 square meter. Even on rocky boulders of riverine forest, among mosses and bryophyte, you see several Graphidaceae competing for its species space. Family Graphidaceae thrive on mangrove trees and on thorny bushes along the seashore. Graphidaceae is even abundant on mountain peaks. Graphidaceae is also very common in man-made habitats such as plantations and home gardens. Several species are even found on bamboo trees. Allographa bambusicola is a new species collected from high-altitude bamboo plants in a cardamom plantation in Sri Lanka and described in 2019. The name of the new species refers to its bambusicolous growth. Graphidaceae is not only found on tree trunks, but also on twigs, branches, tree canopy, and buttress roots. If your hobby is climbing trees, you are a winner. You are a winner to collect some of the rarest species from the canopies. Although most of the tropical countries in whole of Asia have incomplete information on lichen fungi due to lack of experienced taxonomists as well as the lack of basic taxonomic and ecological data for most areas, from the mid-1900s to the present, many new species and many new records were documented from South Asia. Currently, around 1,000 Graphidaceae species have documented for South Asia. Highest number is recorded for the tropical island Sri Lanka. Dearth of information is available for Nepal, Bhutan, Bangladesh, and Maldives, while no data available for Afghanistan. Over 100 new Graphidaceae have been described from South Asia since 80s. Most of these species need to be revised following new generic concepts. Also, type specimens need to be carefully studied to ensure their validity. However, access to these type specimens, most of the type specimens in South Asia, is a challenging problem. Now, I would like to talk about few of our recent findings from Sri Lanka. Recent studies enhance the baseline taxonomic knowledge of lichens of this highly diverse island biome. We were able to document over 300 Graphidaceae species, including about 170 new records from our new collections, and our collections led to the discovery of 37 new species in family Graphidaceae. Further, another 25 new species are awaiting to be described. We are preparing their manuscripts. The most speciose genus for Sri Lanka was identified as Graphis by our research group in 2012. In 2018, however, the genus Graphis was divided into two genera, Graphis and Allographa, by Robert Looking and Klaus Kalb. The total number of species of Allographa and Graphis currently recorded for Sri Lanka is 124, thus surpassing the previously reported numbers for Costa Rica 115, Thailand 115, and Mexico 71. A total of 106 species out of 124 are reported for the first time in the country. This is remarkable as Sri Lanka covers an area of only 65,000 square kilometers, being an island, compared to Thailand with 513,000 kilometers, almost 8 times the size. Biogeographical comparison study was conducted using graphics and allographa to investigate a potential signature of the biotic ferry species interchange. Sorensen pairwise comparison conducted for 4 countries showed Sri Lanka and Costa Rica are more similar, although Thailand and Sri Lanka are found on Asian continent or in paleotropics. One explanation for the higher similarity of Sri Lanka with Costa Rica is that this is a signature of the so-called biotic ferry species interchange. But Sri Lanka was part of the Gondwana until 160 million years and eventually collided with Eurasian plate around 55 million years ago, carrying both Gondwana and endemic elements acting as a biotic ferry. For this hypothesis to hold, species composition in areas of tropical Africa should be similar to those in the neotropics, which is currently not possible to assess due to the lack of data from tropical Africa. We heard from Professor Rosemary yesterday that lichens go back to 400 million years. Their survival story over these years. Nevertheless, molecular clock study shows that graphics and allographa did not diversify until 30 to 50 million years ago, and the age of most species is probably not greater than 5 to 10 million years, meaning that the genera and their species originated long after the Gondwana breakup and even after the collision of the Indian subcontinent with the Eurasian plate. Some taxa currently identified as a species in these graphics and allographa inventories represent species complexes. So one of the best example is Graphyscripta, which is a species that was a cosmopolitan species. In most cases, this species is overlooked. A more likely explanation would be geologically recent mid to long distance dispersal, facilitated by paleoclimatic conditions, combined with the relative proximity of India and Sri Lanka to the African paleotropics as compared to other parts of tropical Asia. I carried out NMS ordination of vegetation types in lichen species space. 146 Graphidaceae species collected from 8 different vegetation types were used for this analysis. Several different environmental parameters were collected and used for this ordination. Ordination explains 61.5 variation by 3 axes. Vegetation types strongly pattern on the ordination, proving that species composition depends on respective vegetation types. First axis showed 33.4% variation, while second one had 17.2. DBH was strongly positively correlated on the first axis, while altitude has a negative correlation. Light intensity was strongly correlated on the second axis. Indicator species analysis, which I carried using 146 species in 8 different vegetation types, showed that 46 species were significant indicators and were exclusive to one vegetation type. 65% of these indicator species were rare species. 49 were extremely rare. In conclusion of NMS and ISA, highest Graphidaceae diversity recorded for the lowland tropical rainforest. Rapid invasive species were found in exotic plantations and had the lowest diversity. 40% of the species recorded were frequently occurred throughout the vegetation types. The study confirmed Graphidaceae, the most speciose family, is useful bioindicators to assess degree of disturbance and ecological continuity in the tropics. I would like to take the last minute of my talk to point out real challenges for lichens in tropical Asia. As mentioned earlier, most of the tropical countries in Asia still have incomplete information on lichen fungi due to lack of experienced taxonomists. Many of the ecosystems are vulnerable to climate change and potentially for desertification. This may alter the species assemblage's ability to survive. Ecosystems that are fragmented, either naturally or as a result of habitat destruction, are likely to be most at risk at climate change-induced species losses. As species are unable to survive, changes in climate may become regionally extinct if unable to disperse, and other species may not be able to travel through the matrix to reach such fragments. Therefore, many species in the region may be driven to extinction while still undescribed. It is very important that these threats and their drivers are dissected and discussed to understand the real threats to biodiversity on a regional and local level, and to nestle this within the global context to implement most desired conservation efforts. Thank you for listening.